I'm the only lowly scientist in this crowd. And so there's a very good thing. Nobody lets me any near patients, so that may be a good thing. And in my job, we're in a way, for better or worse, we're a bunch of people who think about ideas, and sometimes that is important as well. And so when we confront a problem that in pancreatic cancer we do not have targets that we can target with drugs, such as these mutated oncogenes. In pancreatic cancer, it's not just RAS, as he told you, so I make it a bit more simple there. There are about 10 of them that are there, but none of them actually at the present time can be targeted with small molecular drugs. And so if you deal with that type of a problem, how can you eat your cake and, well, have your cake and eat it too, really? So what can you do in such scenarios? And for problems like that, sometimes you need a scientist. And in fact, we found a solution to this problem in principle, and all that finding and discovery happened here at the Wilmot Cancer Center. And that gives us really the opportunity to start thinking about the next level of targeted cancer interventions that we call targeted cancer treatments 2.0. And actually, it turns out it's a relatively simple idea. And that may be the reason that this could be even a good idea. Not all ideas are good. And we are not different from anybody else. We have a lot of bad ideas, and we need to sift out and distinguish. And part of our work is to distinguish the good ideas from the bad ideas. And that's why nobody lets us to treat, touch patients. That's a good thing. But so as a scientist, you are forced to be able to make mistakes. Without mistakes, we don't learn. And so that's why we have to sift out between good and bad ideas. And that's why there is this world of ideas out there that needs to be really taken very seriously. And that's why the scientists really work mostly in a world that is invisible to you, really working on the foundation of buildings, so deep in the ground below the basement. And so when you look at sort of the molecular structure and underpinnings of how cells work, then you discover very quickly that one gene doesn't do a thing. There are about 25,000 genes in cells, and they all work with each other in coordinate networks that create what we call biological responses, biological behavior. For example, if a cell should divide or not divide is all controlled by a vast communication network. And so that argues that these mutated cancer genes that really convert or help convert a normal cell into a cancer cell are not doing their job alone. And what we found is that there is a set of mediator genes that are non-mutated, that are critical for these cancer mutations to generate cancers in people and in other models. And what is also very exciting is that a large diversity of these cancer mutations seems to work through a common family of a smaller number of these mediator genes. And that argues, to a certain extent, that if you would target mediator genes, the right ones in the right circumstances, that you actually could treat cancers in patients extremely well. That also may provide us means to actually treat many patients with highly specialized molecularly targeted treatments in new ways in years down to come. So we're very excited about this, and that's not only that principle seems not only to apply to pancreatic cancers, but to a variety of cancers. And that's why we're even more excited about this whole principal idea. So we think that there is much more to come, and I'm just giving you sort of the very simple surface of those ideas. And so what is really interesting for this morning is that we have a wonderful example that we're working on. And in fact, that one of these mediators is very important in pancreatic cancers. This is a gene called, has a funny name, Plaque8. It doesn't matter what it exactly means, but this gene is related and controls the rate of recycling in the cancer cells. And so what is that? Well, let's start at a simple observation. First, if you look at pancreatic cancer, and this is looking at the cellular level, where in this image here you see normal pancreatic tissue, and this is pancreatic cancer, and this is a ductal cancer. The normal tissue does not express or not make or produce this mediator, but the cancer produces this mediator protein. And what we know is that without this mediator protein, the cancer cannot actually grow. And what we have learned over quite some years is, in fact, that pancreatic cancer cells rely on an activated recycling process. And so what does that mean? So in a way, cells do have discovered millions of years ago what we're discovering right now in our towns, that, well, you have to have garbage removal and you have to have recycling, and this all makes great sense because cells are dynamic structures, and you build things and you have to take down things, and if you have to be, you want to be very efficient at this, and so this is a cycle that turns. And in cancer cells, all these processes of building and taking down are actually working at a much higher speed than in normal cells. And that is a great stress that you can imagine that this is very stressful for these cells. And that probably these cancer cells are experiencing a much higher stress level than normal cells, and that more or less you could think about pushing these stress levels slightly over the edge is an opportunity for intervention and treatment. And we just have to figure out what the sensitive spots for intervention are, and we think about this at the molecular scale. And so the two grad students who actually have done the work in the lab and have discovered that Plaque8 is critical for this recycling process are Conan Kinsey and Vijaya Balakrishnan. And when you actually look at cancer tissue and compare this with normal tissue, you see that Plaque8 expression and also these little green dots here that we could call garbage trucks that collect the garbage in cancer cells. There are many more of these, and than in the normal tissue. And what Plaque8 is actually required for is the emptying of the garbage truck. And so, when you take away Plaque8, the cells basically cannot get rid of their garbage, and the whole system collapses. And we can see that in tissue culture experiments in the laboratory, and we can see that also in models in the mouse, where we remove Plaque8 from the mouse, that the tumors actually cannot progress and collapse. This is also exciting because when we model these diseases in the mouse, we can remove the Plaque8 gene from the mouse without the mouse being actually affected in any health status. So what that shows is that this Plaque8 protein really becomes important to that cell once the cell is under stress. And so theoretically that provides us with a window of opportunity to think about treatment approaches in this type of mechanism. And we're studying right now how many pieces and how many elements belong into such a mechanism to figure out how then to design highly specific interventions that in a few years' time could really go into the clinic and be beneficial to patients. And so in a simple form, when you sort of look at this in a very conceptual way, then normal cells do have a recycling rate, and there's an optimal recycling rate that the cells have to be in order to exist and survive and be healthy. Cancer cells live at an increased recycling rate that is beneficial for their existence. And simply when you target this mechanism, you can basically move this optimum back and to the normal cell optimum. That means normal cells should be still fine, and the cancer cells cannot operate under these conditions. So, and that's how we're starting to think at this level. How can you really find treatments that harm cancer cells but not normal cells? Now, all in the end, this may not work. But I just wanted to give you an example of how we think, how we operate, how we push down in the basement to come up with highly specific new treatments here in Rochester that will, in the end, help you and other patients and people conflicted with cancer. Okay, and so yes, I think this is worth it. I think there are lots of very interesting projects going on here. The group also that is involved in specifically investigating pancreatic cancer at different levels is growing. The support from you has been instrumental to make this all happen, and at that level, it's fantastic how your group is supporting us to make these new ideas and treatments possible. And from that point of view, the collaboration between you and us is essential to drive this forward, because without your help, we would have not been able to collect the data that made us competitive to compete successfully for federal funding. So thank you for that. This has been absolutely terrific. And we know especially the pet narrows have helped us, and I mean, this is just an example for how the community there is, and we thank you guys for being there and working together with us on this very important issue.